I'm here with my group from University of Uyo. We'll be presenting a detailed explanation of two important laboratory techniques, Agarose gel electrophoresis and polyacrylamide gel electrophoresis page. By the end of this session, you will be able to clearly understand how each method works, the application, and why scientists choose one over the other for different types of laboratory analysis.
Have you ever wondered how scientists can see, separate, and study DNA? That's right. From something simple as just a strand of hair or even a glass of water I just drank. That's right, every sip of water carries tiny cells from your mouth, and inside those cells is DNA, the molecule that carries all your genetic traits. The secret lies in a technique called electrophoresis, and today we will show you exactly how it works.
Basic concepts of gel electrophoresis. Gel electrophoresis is a laboratory method that separates molecules such as DNA, RNA, or proteins. We place the samples into a gel and apply electricity. Smaller molecules move further and larger molecules move less. At the end, we see different bands that show molecules of different sizes. This gives a clear way to study how big or small each sample is.
I'm here to talk about the basic concept of electrophoresis, specifically the subtopic that says why the molecules move. The molecules move because they are negatively charged. DNA and RNA have neutral and negative charges. Proteins do not so move, as we well know, but use chemicals like SDS to give them negative charge. When the power of this protein is turned on, these negative charge molecules travel towards the positive side of the gel. The movement helps separate them based on how fast and how accurate they are, depending on the time, speed they use and the rate at which they travel at.
The role of the gel and pore sizes. Firstly, what is a gel? A gel is a medium for separation. The gel usually comes either as agarose or polyacrylamide. It acts as a molecular sleeve. It holds samples in place and allows charged molecules like DNA, RNA, or protein to move under an electric field. The gel slows down movement based on molecular size, while pore sizes is determined by the concentration of gel used. Lower concentration is equal to larger pores, that is good large molecules. Then higher concentration is equal to lower pores, better for small molecules. The importance of pore sizes. It controls the speed and resolution of molecule separation. It ensures smaller molecules that move faster and larger ones move slower, allowing size-based separation. Choosing the right pore size is the key for accurate and efficient analysis.
Agarose is a natural material that comes from seaweed. When heated in buffer and cold, it becomes a firm gel. Agarose gel have larger pores, so they are best for DNA and RNA coding. They can separate fragments that ranges from hundreds to thousands of base pairs. This makes agarose very useful for genetic studies.
Okay. To run an agarose gel. How agarose gel can be used. To run an agarose gel, the agarose powder is first poured into a buffer and heated. After heating, the liquid is then poured into a tray and a comb is added into it to create wells. Upon cooling, the buffer is then placed into the electrophoresis tank, which loads the DNA sample. And then electricity is applied through it, so the DNA can move to the gel. After the run, we use staining dyes to see the DNA bands.
Advantages and uses of agarose gel. Agarose gel is safe, inexpensive, and easy to prepare. That is why many laboratories use it every day. Agarose gel is used to check PCR results, analyze DNA cut by restriction enzymes. It is also used to measure DNA sizes. Agarose gel can also be used to check if DNA is broken or intact because it is simple. Yes, it is simple, and even beginners use it successfully because it is simple. Thank you.
Agarose cannot
separate very small molecules or proteins well. The pores in Agarose are too large to show small size difference. For proteins or small fragments, scientists use pH, which usually stands for polyacrylamide gel electrophoresis. pH has a very small and uniform pores, which gives higher resolutions. It is used when we need more accurate and detailed separations.
Polyacrylamide gels are made from acrylamide and bisacrylamide. Acrylamide forms long chains, and bisacrylamide connects those chains together. This reaction creates a strong gel network with tiny pores. Low gel percentage produces larger pores, while higher percentage forms a tighter matrix with smaller pores. The liquid mixture is poured between glass plates with a comb, and it solidifies to create wells for samples. During electrophoresis, an electric field pulls molecules through the gel. Smaller molecules move faster, while larger ones move more slowly. After staining, separated molecules appear as clear bands that show their different sizes.
Hello everyone, today I'm going to be talking about the SDS page for protein. First of all, the SDS page is the most common type of page that is used to separate protein. The SDS coats proteins, give them a uniform negatively charged. Eating and chemicals unfold the protein so that size could be the only factor. Smaller proteins move further down the gel, while larger protein stays near the wells. After the run, the gel is stained to make the protein band